For the opportunity to present our work on linking phenotypic differences between species to differences in their genomes. So I'm deeply fascinated with this great phenotypic diversity that evolution has produced. And we've seen similar slides in this symposium, this conference before. And I think this is what probably Darwin meant when he wrote about endless forms most beautiful. So in the genomic era, all the species shown here, and of course many more, have sequenced genomes. And this allows us now to address a key question, and that is, well, what are the differences in the genomes that are associated with particular traits that differ among these species? And this is kind of the main question that my lab is interested in. So to link phenotype and genotype, we've developed an association-based approach that we call forward genomics. And this approach focuses on ancestral traits that got changed or lost in independent lineages. And then it conducts a genome-wide screen that considers all genomic regions that overall are well conserved among the species. And we are trying to find those regions that are diverged or even completely lost, specifically in those species that have changed or lost the trait. And on the other hand, they should be conserved in those species where the trait is present. So here's how we do this. Here you have the phylogeny of the species of interest, and imagine we would focus on one genomic region at a time, so typically a few hundred base pairs. Then we can computationally reconstruct all these ancestral nodes here, all these internal nodes in the tree. And with this allows us to now focus at the start and end of every branch, and in a way measure how much of the sequence has been conserved along that branch. So in this toy example, we see that 11 out of 15 bases are identical, so we can say 73% of the sequence was conserved on that branch. And then we can classify all the branches into two groups, into those in green along which the trait was preserved, or along those in red where the trait was changed or lost. And this you can do with parsimony or likelihood. This is what you can use. So now we have those two groups of branches, and we have per branch a sequence conservation value, and those values are independent. That's important. And we can now ask whether the trait loss branches are associated with lower conservation values. So we can use Pearson correlation to quantify this, and if we get a significant p-value, we found an association between this trait and the divergence pattern in this genomic region. Well, now we can march over all conserved regions in the genome, and if you like, generate a macroevolutionary Manhattan plot that you probably know from GWAS. So let's get into a specific example. If you look at this tree here, you see that there are 4 independent mammalian lineages that have adopted a subterranean lifestyle. And this is always associated with eye degeneration and loss of eyesight. So we were interested in what are the genomic changes associated with eye degeneration in these mammals. Forward genomics looks for sequence divergence in conserved genomic regions, and they largely fall into 2 classes. The first class are exons of coding genes, and here we find significant sequence divergence in these subterranean mammals in exons belonging to 208 genes. And interestingly, those 208 genes are highly enriched in eye-related genes, as well, you can see here from these gene ontology or MGI enrichments. The second class are those conserved regions that do not belong to genes, and they often overlap cis-regulatory elements. So here we screened a large set of 350,000 of these conserved non-exonic regions, and we found significant sequence divergence in about 2.7% or 9,300 of them. And we could show that this set of diverged non-coding regions is highly enriched in overlapping regulatory elements that are active in mouse eyes, eye tissues, like functional genomics data of adult retina or lens, are also developing eye tissues. So forward genomics finds hundreds of genes and regulatory elements, or non-coding regions, that many of which have a relationship to eye, that have sequence divergence in these subterranean mammals. So we think that both divergence of genes as well as regulatory elements contributed to eye degeneration in the evolution of these mammals. So the approach that I just explained is unbiased in that it can deal with both genes as well as regulatory elements in pretty much the same way, because all we do is counting nucleotide differences. And while this is on the one hand nice, it also means we're ignoring biological knowledge we actually have about the different types of elements. Specifically, regulatory elements, we know that they consist of binding sites for transcription factors. And we can use the binding specificity of the factor, which you can often describe as such a logo here, to actually predict binding sites in the sequence. So to improve the power to detect relevant divergence in regulatory elements, we developed a method called regulatory element forward genomics, or Reforge for short. And this is based on exactly the same reconstruction framework, but now we're not measuring sequence conservation, but instead we're measuring how well the transcription factor binding affinity is conserved along a branch. And we applied this approach to the same set of conserved non-exonic regions, and this time we found significant binding site divergence in 3,700 of these elements. So this means we found less elements than before, so likely we have improved specificity. And importantly, the smaller set actually has a higher enrichment in overlapping eye regulatory elements. So probably we have higher sensitivity as well. Coming back to genes, we of course know that mutations in coding exons can be synonymous or non-synonymous, but we actually found that many of the diverged genes we looked at before are actually inactivated, and they have accumulated nonsense mutations or frame shifts, so mutations that kill a gene. So to improve the power to detect differences at the gene level, we developed a pipeline to systematically identify genes that are lost. And with this approach, we can now link convergent traits to directly to convergent gene losses. And the particular example I would like to highlight is adaptation to a carnivorous or herbivorous diet in mammals. So we screened for, systematically screened for genes that are preferentially lost in herbivores, as well as genes that are preferentially lost in carnivores. And we found a number of interesting genes for both of these groups. In the interest of time, I will explain the significance of one of them, which I think is really interesting. And this gene is PNLIP-RP1. And this is convergently lost in 8 independent herbivorous mammalian lineages. The function of this gene relates to the digestion of dietary fat, so triglycerides. To do this, you need the enzyme pancreatic lipase. And this enzyme is only active when it binds to a cofactor. Only then it can cleave triglycerides. PNLIP-RP1 has no enzymatic activity, but it can bind and sequester the cofactor. So if you have this gene, or if the activity of this gene inhibits the triglyceride lipase. And this is an effect you can see in mouse knockouts, where enzyme activity is substantially increased, and those mice essentially get fat and have a twofold higher fat mass. So losing PNLIP-RP1 would increase the capacity to digest dietary fat. And we think this could well be an advantage for these herbivores that typically consume a diet that is rather fat poor. So here's a summary. I talked about forward genomics as a kind of a framework to link phenotypic to genomic differences, and I've introduced an unbiased approach as well as 2 versions of it that focus on regulatory elements or gene losses. And with that, I would like to thank the people who actually did the work. I highlighted all those that worked on the project I talked about. Thank you very much.
Questions before coffee?
Nick?
For your underground mammals, why do you think you see eyes and maybe not hypoxia or other groups coming up for which they converged? Also, do they think they have more genes for these, or why do you think the signatures?
It's a good question. I think so. We haven't found any enrichments for lost or diverged genes or regulatory elements that hint towards hypoxia. Likely because you don't really have that many hypoxia genes, right? But you have a whole bunch of photoreceptor lens genes and then a whole machinery, right, for eyes. The other part is likely that hypoxia response is probably more like a gain of function, while losing or degenerating eyes is more like a loss of function, and this is primarily what the method picks up. In the front.
Great talk. I was wondering, all your methods, they kind of work in kind of degeneration of sequence model.
Yeah.
I was wondering, is there any ideas maybe to have like a real adaptive thing where you like a new genetic information or something?
Yeah, I mean, what we're doing right now is, trying to look systematically for something like gene duplications, right, which is in a way a molecular gain of function signature. We have been doing work on screening systematically for parallel amino acid substitutions, which can also be gain of function. But I would like to highlight that, so in general, of course, it is, I think, very hard to infer novel genes or novel regulatory elements entirely from sequence, right? For this, you need to generate functional data, RNA-seq, ATAC-seq, something like this. But I would like to mention the P and lib RP1 example. I think shows that sometimes losing ancestral genetic information can actually be involved in adaptation or gain of a trait, if you like. And we have several other examples found over the years. There's a question over there.
Hi. So you mentioned that for your Forward's genomics approach, you only use conserved regions. And so typically, can you give an indication what percentage of the genome you're looking at then? Yeah. So for the most part, we have been working on mammals. And there it's debated how much is really under conservation, but it's largely something like 5% plus minus a bit. This then depends on how we filter the data. So specifically, we've been using fastcons, which is a widespread method to detect conservation in genome alignments.